Yes, okay, cool. Well, thank you very much. And it's a pleasure to be invited for this talk. And my name is Jérôme Pinti. As Heather just said, I finished or I handed in my PhD last week, and I will defend it at the beginning of December. I did my PhD at the Center for Ocean Life, which is a center within DTU Aqua, which is the National Institute of Aquatic Resources of Denmark. The main difference when you do a PhD in Denmark and I guess most of Europe with the US is that it's for a fixed period of time. Mine was 3 years, so I knew from day one which day I had to hand in my thesis, and that explains why some of what I will present to you is still a bit work in progress, but I will also tell you how it will change in the future. Okay. So before we start, I need to acknowledge absolutely Andy and Thomas, my two supervisors doing this PhD, and most of what you see wouldn't have happened without them. I also want to acknowledge all my co-authors from DTU Aqua and from other institutes, and I will tell you more about their contributions as we get through it. Yes. So for the next 45 minutes or so, first I will tell you about DVM, so Diel Vertical Migrations. I will use this acronym like 10,000 times, and I hope you will know a bit more about it at the end of this talk. And then I will tell you a little bit about game theory, what that is, and why I use this as my main methods for the 3 years of my PhD. And then I will focus on 2 of my 4 PhD chapters. The 2 last ones, one which is about zooplankton size-based nonlinear vertical migrations, and the last one which is linked to the biological carbon pump. So before we start, what is DVM? The DVM is basically a predator avoidance mechanism. So if you think about zooplankton, they have a trade-off. They need to eat and they need to avoid being eaten. And you can think of this trade-off in the water column as to eat, you need to be at the surface if you're a zooplankton because you feed on phytoplankton. But to avoid being eaten, especially by visual predators, you need to be where it's dark because then they can't really see you, then they can't really catch you. And to do that, copepods, zooplankton, and many other organisms came up with a very simple but still very smart solution, is that during the day, they hide at depth where it's very dark and then they can't be seen. And during the night, when it's dark, then they go back up to the surface where they can finally feed without risking to be eaten too much. So if you look at ecograms in the major ocean basins, so what you see here are 4 ecograms from 4 different ocean basins. So on the x-axis, you have the time. On the y-axis, you have the depth. And the color is the backscatter. So the acoustic backscatter is kind of a measure of biomass, but it's not a measure of biomass, but it gives you an indication of where the organisms are in the water column. So you can see that there is one very common pattern within all ocean basins, is that organisms do perform this Diel vertical migration. Here they remain at the surface during nighttime. When the sun rises, they go down, and then they go back up again around sunset before staying up. But despite these similarities, you have a lot of differences. You can have several layers, several deep scattering layers here. You have differences in the depth of the deep scattering layer from like 300 meters here to around 700 meters for the Indian Ocean, etc. And this is because light is not the only driver of vertical migration, but because you need to consider the trophic interactions, as I told you before, because the main driver of DVM is this predator avoidance mechanism. In particular, if you look at this echogram that was taken in the Norwegian fjord by Godot et al. In their paper from 2014, you see that you have first a typical pattern I told you about previously, where organisms stay close to the surface at night, then they go down as the sun sets, and here you see that they seem to be remaining around 80 meters, but suddenly there is a big drop and they go down around 120 meters suddenly. And this is because right around this arrow there, just before noon, in this fjord arrived a big school of predatory fish. So suddenly the predation pressure increased tremendously and the organisms had to find refuge somewhere to decrease even more their chances of being eaten. and they found their refuge where it's dark, where it's darker, so at depth. Then when the sun started to set again, they could go back to the surface and resume their normal patterns. So I guess that from this, I could convince you that there is a need to consider trophic interactions to model Diel vertical migration patterns. And to do that, the main tool that I used in my PhD was to consider, is that working? Yes, game theory. So game theory is something, it's a branch of mathematics, let's say, that was invented by John Nash here, an American scientist, I guess most of you know him. And we can define game theory as the study of mathematical models of conflict and cooperation between intelligent and rational decision makers. What I was not interested in cooperation at all during my PhD, because all I cared about was conflict and predator-prey interactions, why organisms want to eat each other. But you can have game theory studies where you need to cooperate with each other, for example, in a social settings of monkeys or whatever you can have. Yes, I think the intelligent, rational decision makers here is important to note, and it's hard to justify that a fish or a zooplankton is an intelligent and rational decision maker, but we just argued that fish and zooplankton evolved towards the best strategy for them and the one that optimizes their fitness at most. And this equilibrium, this optimal solution that we want, its optimal distribution, is what we call the Nash equilibrium. And it's the point in which no player has anything to gain by changing only its own strategy. So if you consider that the players are the fish or the zooplankton or any marine organisms that can perform a certain migration pattern, then if it changes its migration patterns, it cannot increase its own strategy, or it cannot increase its fitness, sorry, not its strategy. So if the Nash equilibrium tells you first go at the surface, then go at depth, then go back to the surface, if you change this pattern, there is no way you will get a higher reward. And this is what we call the Nash equilibrium, and this is what I looked for in all of my chapters. Yes. Then how to find the Nash equilibrium. So this is the only technical part. I'm sorry for the 2 slides with the equations, but there won't be any more after, I promise. So if you, for simplicity, I only considered Diel vertical migration as a 2-step process. So the position during nighttime and the position during daytime. And I didn't consider the shape of the descent, ascent, or the timing or something like that. And then I can summarize the strategy that organisms do, meaning the strategy is the position Nij, I being the position in the water column at night and J being the position in the water column during daytime. I can summarize it in the form of a matrix here, where all the little Nij would be the fraction of organisms that follow this strategy. For example, N11 would be the fraction of organisms that stay at the surface at all times, and Kk is the fraction of organisms that stay at depth at all time, etc. So if you stay in the diagonal, you don't migrate at all. You're doing the residency in the layer that you have here. And here are the extreme migrations between the surface and the depth during day and night, vice versa. So for each of these strategies, what I can do is that I can assign it a fitness, so a measure of how well the organism is doing. And this fitness is simply defined as growth over mortality, which is also called the Gilliam's rule. And this fitness, Nij, depends on the fraction of organisms that follow it. So if you have a lot of organisms following this strategy, maybe it will attract more predators, then the fitness value of assigned to this strategy will decrease. But if you're alone doing that strategy, maybe you will have more chances of being eaten by the predators that are present in that water layer at the same time as you, etc. So this fitness that I assign to each strategy is dependent on the fraction of organisms that follow each strategy, but also on the fraction of prey and predators that follow this strategy, or that are at least in the same water layers as the others. So once I have this fitness measure, I can use the an algorithm called the replicator equation to find the equilibrium distribution of organisms in the water column. So basically, I assume at each time step that the relative fraction of organisms following a specific strategy grows according to proportionally to the fitness that is assigned to this strategy. And then in the end, I renormalized just to ensure that the sum of the Nij is always 1 and that I don't have an increase or decrease in a population. I run this equilibrium until steady state, and then at steady state, I have my Nash equilibrium. And we can say that it's in some way it is a flaky algorithm because there is no formally no certainty that you will reach any equilibrium, and there is no certainty that depending on the initial conditions, you will not find several Nash equilibriums. And in everything, in all of the following, I was lucky enough that there was never several Nash equilibrium and that I could always reach this stable point that interests us. So for example, one example of distribution that you would have then at equilibrium is this. You have the position at night on the lines, the position of the day during on the columns. And you can see that most of the organisms then in this configuration would not migrate. They stay always in their water layer. Many are staying all the time around 300 meters. Some are staying all the time close to the surface. And if you look at these fractions of organisms around here, they stay during the day around 200 meters, and during the night, they stay around 100 meters, meaning that these guys are performing DVM. So in summary, in my to run these methods, I need to define a food web of interest with several populations. So here I gave you only one population, but I can have 2, 3, or how many I want. Then for each of these populations, I need to define a fitness measure, which is growth over mortality. This growth and mortality term will depend on the interactions between the prey and the predators, with the light levels that they can have, or whatever I want, basically. And finally, I need to use the replicator equation, which is this game theoretical algorithm, to find the optimal patterns of all components of the food web. Yes. So my first example that I will tell you here is in the California current. I don't know if you know this study made by Oman and Romania in 2016 around the California current. So here you have San Diego, around here you have Santa Barbara. And what they did is that they did 5 different transects. And in each of these transects, they investigated the Diel Vertical Migration patterns of copepods of different sizes. So they basically took, with BP2 nets and plankton nets, they took plankton samples, zooplankton cycle, they size distributed it, and then they looked at the size distribution of all organisms and the depth distribution of all organisms according to their size range during day and during night, so that they could see what is the distribution of these organisms. And you can see that from cycle 1 to cycle 5, you have a clear gradient between clear and oligotrophic water to more turbid and darker waters. Yeah. So their results, then you can summarize it into 5 plots where you have on the y-axis the observed depth or mean depth during the day. And here on the x-axis, you have the size of the organisms between 0.1 and 1 centimeter. And in red, you would have the day distribution of organisms, and in blue, the night distribution of organisms. And here, what is striking is that the small organisms and the big organisms, they don't migrate. They always stay at the same depth. Small organisms are staying closer to the surface than the bigger one that stay always at depth. And it's only in between that you can see that DVM emerges. And this is for the clearer cycle that they did, the clearer place. But as you go closer to the shore, you see that this pattern holds, but it becomes closer to the surface with a smaller migration window here and here, even smaller, but still no migration for the small ones and no migration for the big ones. And the simple way to explain that in a way is to explain it as, well, small organisms, they are so small that they can't really be eaten by fish and visual predators. So basically they don't care where in the water column they are, they can stay always at the same depth. The bigger organisms, they are so big that even at night, they would be able to be seen by visual predators, and then they need to remain at depth all the time. So it's only in between that you have this emergent trade-offs that tells you, well, during the day, you are too big that you would be visible by fish and you need to hide. But during the night, it is safe enough that you can go up to feed in more nutrient-rich and more phytoplankton-rich waters. And our idea with this game theoretic method was to see, like, well, this is nicely explained in idea, but can the math actually justify and validate this hypothesis? So to do that, I applied my methods. First, I created a food web where I had fixed phytoplankton and microzooplankton at the surface, distributed according to the mixed layer depth. Then I had a big food web of copepods that were divided according to their size classes from 10⁻⁻1 to 1 centimeter. In the end, I ended up with 50 different size classes of copepods, and I also divided the copepods according to their feeding modes between ambush and active copepods to understand if there was any significant difference between these two feeding modes. And I can tell you that already you have many more active copepods in the water column than ambush copepods, and ambush copepods tend to be smaller than active copepods. So in the size range that we have, around 0.1 centimeter, you have approximately 60% of the copepods are active copepods. And when you get to above, I guess, 0.4, 0.5 centimeters, 100% of the copepods are active copepods, and you have literally no big ambush predators. And then in the end, for closure, I have fish predators, and all these organisms can produce detritus that sinks, but can then be uptaken again by ambush copepods. And here you see a few arrows for 3 boxes, but in reality, you have many more arrows, and all copepods are allowed to feed on all of them according to size preference functions and a mechanistic description of their encounter rates, movement, etc. So more details can be found in this paper here. If you look then back at the results that Mark Romain and Oman and Jean-Baptiste Romanian had in their paper, that was this that I just showed you before. And my results from what I predicted with my model is that. So what you can see already is that the global pattern holds, like you still have this migration envelope that goes, that gets shallower as you go closer to shore, and you still have the residency at the surface and at depth for the big and for the small and big zooplankton. And of course, you could argue that this is not exactly the same pattern as this, and that you still have a lot of differences, but we didn't try to tweak the model so much as to reproduce precisely this pattern. The idea was just to see if we could reproduce the trend and if this nonlinear pattern could be explained by game theoretic principles, and it could. So that was the main satisfaction and the main point of this study. And then, as I told you before, we have the active and the passive copepods that are in this model, so we can look at the difference between the two. And that's it. You have the dashed lines represents the ambush copepods, and the plain lines represent the active copepods. And what you can see is that most of the migration envelope is due to these active copepods. The ambush copepods are literally staying almost all the time at the surface. And this is something that I had no idea about, and Thomas didn't have any idea about either. And I tried to look in the literature, and I didn't find really any evidence of difference between active and ambush copepods in Diel vertical migrations. You have a lot of reports that small ambush copepods, like Oitona, I think they're called, they remain in the water column everywhere without migrating, but that was not really explained. But when I went and looked back in the species-specific Diel vertical migration patterns, then these results seem to hold, and it seemed that all the active copepods were indeed performing vertical migrations, while the ambush predators tended to be more steady and remaining where they are. Okay. Then once we had these results, we were very satisfied, and we wanted to know if this game theoretic method could hold and explain more exotic patterns. And when I mean exotic, I don't know how exotic it is to you, but I'm talking about Seattle. It's this closed, this little fjord here, or bay, it's a fjord, here it's connected to the sea by here. It's called the Dabob Bay. And in this place, we consistently witness a reverse Diel vertical migration. And if you look at the main trophic assemblage in that fjord, you have small copepods which perform reverse vertical migration, meaning that they stay close to the surface in broad daylight during the day, but they migrate down during the night. And you also have ketognaths, which are big tactile predators, big, like up to a few centimeters max, but a big tactile predators, in our size class that are performing normal vertical migration patterns. And finally, you also have fish, but we know nothing about their vertical migration patterns with the studies that we have. And the question is like, okay, can we actually model reverse vertical migrations? And the results was surprisingly, yes, we can. So what I did to simply mimic the fact that I had ketognaths in my system is that I just increased the size range of my zooplankton from 1 to 1.5 centimeters, saying that we have actually a lot of big tactile predators. And these tactile predators are performing normal vertical migration, staying at depth during the day and closer to the surface during the night. The smaller copepods here, they don't migrate, but we can see that the intermediate copepods are performing reverse vertical migration, staying closer to the surface during the day and migrating during the night. And again, we could have tried to tweak the model parameters and the size preference functions to have the smallest copepods perform reverse vertical migration, but that was not really the point of the study. And we were very happy to have actually reverse vertical migration. And to our knowledge, it's the first time that we were able to model reverse vertical migration from simply a mechanistic model. And for completeness, I can show you the vertical migration patterns of the fish. So green is just the phytoplankton distribution that is fixed with a mixed layer depth around like 30 meters. And the fish are kind of scattered during the night, and then they go closer to the surface during the day with something that looks like a bimodal distribution. So that was my first or my third PhD chapter. And the last chapter, well, I decided to get bigger and move deeper, meaning I will focus on mesopelagic fish in a complete pelagic water column. So this is the typical pelagic food web that I decided to go to, because what I wanted to do was to understand what would be the carbon export that is derived from zooplankton and fish. So the food web I have is that I still have a pool of phytoplankton and microzooplankton that can feed mesozooplankton and macrozooplankton. In turn, those two feed forage fish, mesopelagic fish, and tactile predators, and the large pelagic fish can feed at all times on these three. So here I consider that the vertical migrators are these 5 groups. You can tell me that large pelagic fish should be able to migrate, and that's true. But it made the model more difficult to run because there was no real closure term. So what I decided to do was to have a uniformly distributed top predator and large pelagic fish in the water column. And that's not too far off, because actually most large pelagic fish go so fast in the water column that they can really skim through it several times a day. So I think it's okay to consider that they are uniformly distributed in the water column. And then all these organisms create detritus that can sink and get uptaken again by the zooplankton. So my goal with this project was to compute the active carbon export estimates on the whole globe. When I say on the whole globe, I quickly simplified my problem. I didn't consider the extreme latitudes because, as I told you before, the fitness measure I chose with gross over mortality. And when you consider a simple measure, you only consider it over a daily cycle, so it's kind of an instantaneous fitness. And as soon as you consider latitudes which are higher than roughly that and that, you need to take into account seasonality and the fact that animals have an annual routine. Copepods can undergo seasonal vertical migrations, etc. So just considering what they do during one day as the ground truth without any yearly cycle is not good enough. Also, I didn't consider the coast because I wanted to have a focus on mesopelagic fish. So if you don't have mesopelagic fish, then it's not really worth it. So I discarded all areas shallower than 500 meters. Finally, in the project I told you about before, the only environmental driver was the light levels at the surface and attenuating during the day, and attenuating at night and as we go to depth. But here I also considered temperature and oxygen. When you go to this great depth, I think temperature really matters because it changes the metabolic rate of organisms. So if you decrease your metabolic rate, it's a way to conserve more energy. So having low temperatures when you don't want to feed is also good. But it also decreases your SDA, so specific dynamic action, meaning that you can't move as fast or you can't digest as fast, etc. So if you catch organisms, it may not be a good idea to be in low temperatures if you can't digest them. I also consider oxygen, which is said to be very important for deep scattering layer depth prediction. If you have, for example, a big oxygen minimum zone, you can't really, some organisms can, but for example, fish wouldn't be able to migrate through it. They are too small. And an OMZ really has the power to create a barrier and to decouple like two places of the water column. And finally, the inputs that I needed are the biomasses of all the functional groups I considered, because my model is not dynamic. In terms of population dynamics, I only consider their behavior, and I have fixed population sizes. So the biomasses that I have are those. So here you can see the estimates of phytoplankton resources. Intermediate is mesozooplankton, largest macrozooplankton, forage fish, mesopelagic fish, and top predator. So these three are outputs of the cobalt model, and forage fish and top predatory fish are outputs of the FISTI model, which all five were provided by Colin Petrick, one of my co-authors, and the FISTI model is directly coupled to the cobalt model of Charlie Stock. So you should have some kind of logic between these five runs. Then the mesopelagic fish biomass is kind of different. It was given to me by Roland Proux and Andrew Brierley from Scotland. And basically, when I contacted him, I saw two of his papers of Roland Proux. I don't know if you're familiar with them, but one of them is just one global estimate of mesopelagic fish biomass, like how many mesopelagic fish are there in the whole world. And the other paper is because there has been estimates before, but Roland tried to correct the estimate to say that, well, we suddenly said that there were many more mesopelagic fish than previously believed, but maybe we need to decrease this estimate a little bit because all the acoustic backscatter that we get back from mesopelagic depth is not only due to mesopelagic fish, but it can also be due to other things such as siphonophores, etc. So this estimate tried to kind of account for these differences. And then the second paper that Roland Proux published was, I think, in the ICS Journal of Marine Sciences, just a global estimate of mesopelagic fish abundance, but a relative estimate. So he had a map divided into oceanic provinces, and in each of these provinces he was saying like, well, based on the backscatter, here we are likely to have a lot of mesopelagic fish, here we're likely to have less, etc. And then because I was looking for a map with absolute value, I just contacted him and said like, hey, can we just couple this global estimate with this relative abundance to have one global abundance? And this is the map that we have here. Yes. So once I had all these biomasses estimate, the first step was to find the distribution of organisms in the water column throughout the world. So for example, if I give you an example here around a bit north of Hawaii, the Nash equilibrium of the system is something like that, meaning that mesozooplankton are kind of at the surface all the time, maybe performing a very slight reverse vertical migration, but not very clear. Whereas macrozooplankton are performing a clear deviant pattern. So on the right side, sorry, I didn't explain that before. On the right side, you have the day distribution, and on the left side, you have the night distribution. And here you can see that the organisms are kind of spread around 300 meters during the day, and they go back to the surface with a very narrow distribution around maybe 50 meters during the night. The forage fish, they stay close to the surface at all times, and that's basically a general feature of forage fish in my model, and I guess in the world, that they stay close to the surface all the time. Mesopelagic fish, on the other hand, they were really migrating between like around 600 meters and to the surface. And it's interesting to note that their optimal distribution during the day is actually a bimodal distribution with a small peak here and then another peak there, and also a bimodal distribution during the night. Finally, jellyfish were also performing Diel vertical migration patterns. So I want to give you another example, for example, in the Indian Ocean. So this is what the distribution would look like. Again, mesozooplankton do not really migrate. Macrozooplankton migrate much more. Also, again, around 400 meters during the night, back to the surface during the day. Forage fish, as always, close to the surface. Mesopelagic fish also having a clear DVM pattern, but this time only a unimodal distribution, and they seem to have a much deeper distribution during the day, around 600 meters and up to like 800 meters before going back to the surface at night, and jellyfish also performing DVM. So once I have done this, I can compute the optimal distribution patterns basically in all places of the globe, and I can look at what is the deep scattering layer depth in the entire globe. And the deep scattering layer depth, I took it as the maximum distribution, the peak distribution of mesopelagic fish during the day. And this is the map that you can see here. So all of this is the peak mesopelagic fish by abundance during the day. So kind of the depth of the deep scattering layer. Overlaid on top here is the data of the deep scattering layer depth from Clivier and Nile in 2016 from the Malaspina expedition. So the Malaspina expedition, for those of you who don't know it, was a circumglobal expedition that was really focused on mesopelagic fish, trying to understand their biomass, their vertical migration pattern, their trophic transfer efficiency, etc. And that's the kind of the study that launched this revival of mesopelagic fish with the new estimates of more than one order of magnitude higher biomass, etc. And what you can see is that this vertical migration, this DSL, deep scattering layer depths that I predict, only based on mechanistic principles, are actually fairly close to what was observed. In the I was very satisfied, especially in the Atlantic Ocean. They fit very well to what we observe. In the Pacific, you have a bit more differences, but you can see that it's shallower where it should be shallower, then it gets deeper where it should get deeper, and it re-gets shallower where it should get shallower, etc. Kind of the only place where you have something which is maybe not so satisfying is in the extreme latitudes, like around here and in the Southern Ocean, where the vertical, the DSL depth should be deeper than it actually is. And that may be related to the fact that I don't consider seasonality once again. So once I have computed that, as I told you, everything is based on mechanistic principles and trophic interactions. So I can try to understand how much each, I have exactly the value of how much each organism eats each organism. So I can look at the organic carbon flow like this. And you can see that phytoplankton then feed a lot mesozooplankton, which then feed forage fish and large pelagic fish, which should a normal decrease in size again. And all these organisms are creating detritus at a certain rate that then goes in the fecal pellet pool. I wouldn't look too much into this number yet, because as I told you, my PhD was only 3 years, and this run kind of arrived slightly too late. But I made new estimates because, for example, I was not satisfied with this because the carbon budget is not balanced. It's normal that it's not balanced because in my my model relies primarily on behavior, and there is no, it's just not in the philosophy and how I set up my model to have a carbon budget, a balanced carbon budget. But if you look at it, then it's still not satisfying that if you add up how much zooplankton eat, for example, it is less than how much they get eaten, excrete, and respire, which is not pictured here. So I did another run where I corrected for these errors to have something more balanced. And the results compared to what I will show you after don't change much, and the deep scattering layer depth, etc., are still robust. It's just these tiny parameters that change how much the organisms are eating. So once you have that, you know how much carbon sinks, etc. So you can look at how much fecal pellets sink below the euphotic zone. So here you have in milligrams of carbon per day, how much fecal pellets sink at each point in the ocean. And for my global model, this results in a flux of 4.8 petagrams of carbon per year in total. And if you don't know how to relate it to, here are 4 other studies that looked at it. And it varies between 3.4 to more than 10 for the estimates. So first of all, I was happy to be in the range of what people came up with. But second, you can see, if you look a bit deeper, you see that most of the estimates are clustered between 6 and 9 petagrams of carbon per year, which is what seems to be the convergent number these days. And especially these studies, you can download the model very easily online. That's Tim DeVries that did it with David Siegel that I visited in California, 2 of my co-authors. And you can download this little model online, and you can look at how much carbon sinks below the euphotic zone everywhere on the globe. So what I did was that I took that model, I just cut down all the areas that I didn't have in my model, so the high latitudes, the coast, etc. And I had this number of 9.1 goes back to 6 petagrams of carbon per year, whereas what I had was 4.8. And I think it's not too far off. And the main difference could be explained by the fact that in my model, I don't consider the sinking of phytoplankton and aggregates. So they can account for a substantial proportion of the sinking flux. This paper said 13%. Some people argue it could be higher, but I think that explains the difference. And also, I told you before, I changed a bit the parameter values to have different values here. And the revised estimate, I don't have this pretty figure yet with the revised estimate, but the global revised estimate is more around 5.5 than the 4.8 I had before. So I also converge more to what the most recent studies show. Then I can also compare what is sinking below any depth, because I basically have the depth profile everywhere of the fecal pellet sinking flux everywhere on the globe at each depth. I can compare it with the sediment traps data. In particular, there is this Lutz et al. 2007 database I can compare it with. I was kind of happy to see that there is no real bias. I mean, you have a spray, I mean, let's be honest about it, but the sediment trap data are notoriously messy, and it's kind of satisfying to see that there is no big bias in my results with what was observed in the field. And you don't have any bias in plus or minus, but you can see that if you look at geographic and regional breakout, you don't have a bias either. So this map is maybe a bit hard to understand, but each circle is sediment trap place. The size of the circle is related to the depth of the sediment trap. So if it's round, it's close to the surface, and if it's big, it's a very deep sediment trap. And the color of the circle is the difference between the computed and the observed particle flux. So each circle is one of the points that went into this histogram here. And what you can see basically is that you don't have any circle that is any place where all the circles are red or all the circles are blue. So the spread is kind of evenly distributed among the globe, which is also satisfying. So once I had validated my model with that, I could look at how much carbon is excreted below the euphotic zone, because up to now, I only told you about passive flux of fecal pellets from the surface to below the euphotic zone. But I can also look at how much carbon is excreted and respired below the euphotic zone. So that's what I did here. If you look at the excretion of organisms below the euphotic zones, it's mainly due to macro zooplankton and mesopelagic fish. And the total number here is 0.6 petagrams of carbon that is excreted below the euphotic zone. And you can add to this the total respiration of migrants below the euphotic zone, and that's 1.0 petagrams of carbon per year. So in total, with my functional food web, what I have of carbon going below the euphotic zone is 1 of this respiration, 0.6 of the excretion, of 4.8 of the sinking flux, and it's a total of 6.4 petagrams of carbon per year that ends up below the euphotic zone. So this was already very interesting and very satisfying, but it didn't give me an indication of how long the carbon was stored or how efficient each pathway was at sequestrating carbon, because we always say that mesopelagic fish should be very good carbon exporters because they directly bring carbon very deep when they eat at the surface before excreting it at depth, etc. So what we need to do is we need to consider how the water moves in the ocean. For example, if you excrete carbon very deep in the Pacific Ocean, it can take hundreds of years because before this carbon goes back to the surface. Whereas if a copepod respire the carbon in the water which is very close to the surface or in an upwelling area, etc., it can go back to the surface very quickly and then being put back in the atmosphere as quickly. So that wouldn't be a very efficient storage pathway. So the solution that I had for that was to rely on an ocean circulation model that was developed by Tim de Vries, and that relies on a transport matrix. It's called the OSIM, the Ocean Circulation Inverse Model. And basically what I do is that I have the source terms of carbon everywhere around the globe. So the source terms are either the respiration of the animals or the bacterial degradation of fecal pellets that are produced by the animals. So it's the bacterial respiration due to the fecal pellets. So when they turn DOC into DIC, dissolved inorganic carbon. And then here are the results. So if you look at the animal respiration, you have the rates here in petagrams of carbon per year. You can see that zooplankton respire a lot more than the other organisms, but they don't store a lot of carbon, especially mesozooplankton. They only sequester 0.2 petagrams of carbon per year. And that's because they are always very close to the surface, so their carbon just goes back to the atmosphere very quickly. If you divide this sequestration number by the rate at which they produce this carbon, this DIC, you get a year, which is a sequestration time, and it's measure the time scale at which the carbon is sequestered. And then you can indeed see that for mesozooplankton, the time scale is very low, so this pathway is actually quite poor. For the mesopelagic fish, on the other hand, their sequestration time is quite good, and large pelagic fish, as I said that they are artificially distributed through the water column, their sequestration time is also very good. And in total, I have a total sequestration because of the animal respiration of 124 petagrams of carbon per year. With my revised estimates, the absolute sequestration time kind of remained the same, but I have slightly higher rates and slightly higher sequestration, both for the animal respiration and the bacterial degradation. So here are the results to the bacterial degradations of my fecal pellets. So here are the rates. They are higher than for the respiration, and the sequestration is also much higher. And that's normal because the respiration always stays at the depth at which it is respired before being affected by currents, etc. Whereas the bacterial degradation of fecal pellets, it doesn't happen only at the depth at which the fecal pellets are released, but all the way until the fecal pellet is either completely degraded or reaches the seafloor, where then I assume that it gets respired. So this carbon then gets respired at generally much deeper than the respiration. And you can see that mesopelagic fish and especially forage fish amount for almost 100 petagrams of carbon each of sequestration, with very high sequestration time scale, more than 200 years, which is much more than the mesozooplankton, even though the meso and macrozooplankton sequester more carbon in absolute. And this was very an interesting result because it was the first global estimates of how much carbon is actually sequestered thanks to mesopelagic fish and forage fish. And in my revised estimate, again, it's a bit more than 150 for both. So it's also going higher up. And with this, I thank you for your attention, and I think we can go to the questions.
Great, thank you very much. Can you see the chat right now?
No.
Oh, sorry, it was, okay, so one question from the end. Can you comment a bit on your ability to identify Nash equilibria? It seems like the equilibrium would only apply to specific conditions. Example, metabolic reserves, time of year, feeding status, predation pressure. And there should be many more different equilibria rather than one universal one.
Yes, I mean, each time you change the conditions of a system, you will change the Nash equilibrium. And so this, for example, is one example for this Indian Ocean here. So for this place, I have fixed biomass. There is a fixed oxygen concentration through the water column, fixed temperature, fixed light level. The only difference is between the day and night positions. And you can, in the mathematical way Nash equilibrium are working, you are not obliged to have only one Nash equilibrium, you can have several. And I looked for that to know if there would be more. And luckily enough, I never had this issue in my models.
Any other questions for Jérôme?
I could see Suzanne writing in the
Great.
chat.
Yeah.
How much would the vertical distribution change if you had a large increase in water temperature? So I didn't look at that, but that's something very interesting. And if I try to guesstimate that, I would say that it also depends a lot on the oxygen concentration. I think oxygen is one of the major players in like the DSL depth, and it can really constrain organism from going deeper and higher up. So I think in general, what we can expect in the future would be that organisms will get shallower and shallower, because you would have warmer water, meaning that the bacterial degradation rate would increase, lowering the oxygen, making the oxygen minimum zone more intense and more shallow, meaning that the organisms themselves need to go higher up to be able to breathe correctly, etc.
Thank you, Jérôme. That was a nice talk. And what I'm trying to get a sense of is, you obviously had to define your conditions, but if you make even a dramatic change, what is your gut feeling? Would these vertical distributions change by 20 meters, or could the patterns totally change? Okay.
I think you can reach tipping points. So what I did here, I did a small sensitivity analysis on this where I changed all the rates, but not the environmental driver. That's actually a good idea for maybe later to look at how temperatures impact this. And so far, it looks like the results I have with my definition of the rates, which may be good or maybe a bit off, I don't know. It seems fairly robust, so I don't think we will reach with my model any tipping points that would completely change the patterns. Yes.
I have a question for you on those graphs where you had the red and blue lines of distribution during the day and distribution during the night.
Yes.
It seemed like there was a yes, those kinds.
This one or the one before?
Maybe earlier one. Yes. So it seems like there's a really steep, maybe like inflection point at like around 0.8 or something. Is there something mechanistically that happens there that's shifting things?
No, there is no real difference between the others. And again, it can be where you have this trade-off between different, like eating and getting eaten, and suddenly you get on the other side of this trade-off where it's bad. But all these organisms here, they are really parameterized the same way. It's only size functions, and only size-based rates and functions and visual rates and everything. So to run the model with like 10 or 50, or here I ended up with 50 size classes, but to run it with 10 or 100 size classes is just changing the numbers, and the parameters always adapt to these size-based rules. I can see a question from Trisha Thibodeau in the chat. I'm curious what rates you're using to estimate zooplankton excretion and respiration, given the diversity of zooplankton and their various physiologic rates. Yeah, that's a very good question. So actually, then it allows me to give a bit of background from the Center for Ocean Life. So the Center for Ocean Life was, when it was funded originally, like the pitching idea was to get rid of taxonomy, which is maybe a harsh way to say it, but it's true. And to consider, instead of consider the diversity of organisms that are in the sea, to focus on their common points and to adopt a trait-based approach to those. So you have a few master traits that would regulate all the rates of the organisms, primarily size being the first one of them. And you have a lot of papers that exist on the relationship between size and all the different rates that you have. And one of the professors of the Center for Ocean Life, Ken Anderson, just published a book on that. And he has all these rates that depend on size for zooplankton and fish, etc. And I mainly used those. So for the zooplankton excretion rates specifically, it's very, I didn't use a fixed rate. It's just a fixed fraction of what the organism ingests. And the respiration, this is the standard metabolic cost that is given by this size-based rules. And I can say that all the results that I gave you and all the results that I showed are only based on this standard metabolic rate, so the basal respiration that the animals have. And I plan for the future to consider the SDA, so the specific dynamic action. So basically all the respiration of organisms, which would first of all allow me to have a carbon budget which is balanced, and then allow me to consider all the respiration of organisms, and will increase a lot, like 2 or 3-fold at least, the sequestration of carbon due to the animal respiration pathway. You're welcome. Yes, I don't know if anyone has any other question.
Any other questions for Jérôme? If not, then let's join in thanking Jérôme for a great presentation. Postdocs and grad students stay on the line and we will have a discussion with Jérôme. Thank you all.
Thank you.